Hello, my name is Keir Morse, a PhD student at California Botanic Garden. I'm studying the systematics of the genus Malacothamnus. Today I'm going to give you a bit of an introduction to the genus, tell you why I'm studying the genus, and give you a look at some of my results. Do note that this is preliminary and there's still a whole lot of work to do. The genus Malacothamnus is in the mallow family, Malvaceae. And it's most well known for being a fire follower and being problematic taxonomically. There are 37 taxa described. Of the treatments currently in use, there are 11 to 28 taxa recognized. Malacothamnus can sometimes be confused with Sphaerosia. Malacothamnus all have white to pink flowers. However, some Sphaerosias also have pink flowers. Most Sphaerosias can be distinguished from Malacothamnus based on having orange flowers. When you have either pink flowers or no flowers, you can distinguish the two genera based on fruit. Malacothamnus can be distinguished from Sphaerosia based on having fully dehiscent carpels split into two halves and by not being net veined on the carpels. Sphaerosia, however, the carpels are only partially dehiscent and they're net veined. Malacothamnus primarily occurs in the California floristic province, ranging from near sea level all the way up to 9,000 feet on the east side of the Sierras. There are also some disjunct populations out in Arizona. There are shrubs ranging in height from 3 to 18 feet. Most are typically only 3 to 7 feet tall, but some species get much larger than others. Inflorescences are generally classified as head-like, spike-like, or panicle-like. However, these are kind of fuzzy categories and they can fade into each other. In many cases, there are populations with both spike-like and panicle-like plants growing together. Leaves between species can vary both in shape and in degree of pubescence. However, some species have leaves that look almost identical to each other. These two species grow almost side by side, and vegetatively, you can't really tell them apart. Leaves in a single taxon can be quite variable. You can see the two on the left. These are from plants that were growing side by side and have quite a bit of different shape to them. Malacothamnus generally have two sets of leaves each year as well. On the left, you can see winter-spring leaves, which are larger and thinner. On the right, you see fall-summer leaves, which are thicker and smaller. Here I've shown the same plant with both winter and spring leaves on the left and with summer, fall leaves on the right. Observations at California Botanic Garden showed that pretty much every species drops their leaves around July, replacing these winter leaves with the smaller summer leaves. Malacothamnus have two types of trichomes, the stellate trichomes, which are these white branched ones, and simple glandular trichomes. Typically, the simple glandular trichomes are much, much smaller than the stellate trichomes. In this particular species and a few others, the glandular trichomes are much larger, and this can be really useful in distinguishing these taxa. Here I've shown a comparison of stems of various taxa, and you can note that the size and density of trichomes can be very useful in distinguishing these taxa. Likewise, the trichomes can be very useful in the calyx. Note the photo in the bottom center, Malacothamnus gracilis. This is the one with the reddish glandular hairs, which is quite distinctive. The calyx and associated calyx bracts are some of the most important characters in distinguishing taxa within the genus. Here I've highlighted the calyx bracts with yellow arrows. Calyx bracts are a whorl of 3 bracts beneath the calyx. Collectively, these are called the epicalyx. The size and shape of the calyx bracts relative to the calyx is very useful in distinguishing taxa. Another type of bract useful in distinguishing taxa are stipular bracts. On the right, I've circled a cluster of flowers. On the left, I've dissected this cluster. You can see labeled in here, these stipular bracts are essentially bracts that are a modified stipule. In some species, these are greatly modified in size and shape, which makes them very distinct. Malacothamnus are fire followers. Germinating after fires, they generally live 3 to 20 years. Some may live longer. It's unknown how long they actually may live, but usually they're all dead by the time the next fire comes around. Fire return intervals are 20 to 120 years. Based on this, seeds are likely viable for 100 plus years. In the bottom photo, you can see the boundary of a burn area of pinyon pine woodland. On the left side, you can see pinyon pines are all alive. There's no sign of Malacothamnus anywhere in this unburned area. On the right side, this is 2 years after the fire, Malacothamnus is the dominant species. There's no sign of any living pinyon pines in this area. This photo shows the first June after this fire, seedlings coming up. There's about 50 plants in this photo. Probably only a few of those will actually survive as they outcompete each other. This is the same area the second year after the fire. At this point, Malacothamnus is the dominant species in this area. How long Malacothamnus will persist in this area or how long it'll remain a dominant species is something that is unknown. I have gone through some areas recently as old as 20 years old that still have some sparse Malacothamnus of the same species. There are two main types of seed dormancy related to fire falling. There's physical dormancy, where the burning breaks through the seed coat, allowing water in, and chemical dormancy, where there's a chemical reaction with the chemicals from smoke. The seed bank at California Botanic Garden tests various treatments for their effectiveness in causing germination in seeds. These photos show two germination techniques that the seed bank has tried to employ. On the left, you see clipping, where they have removed part of the seed coat with a nail clipper or a razor. This essentially allows water to penetrate the seed coat. On the right, they just planted some seeds, put some sticks on top, and lit them on fire to see what would happen. Shown here is percent germination of Malacothamnus seeds based on treatment. You can see on the far right, with no treatment, very few germinated. You can see in the center, clipping was the most effective treatment. This shows that it was a physical dormancy in the seed and not a chemical dormancy. Smoke water was not needed to get these to germinate. This is occasionally evident in roadside gratings where seeds have been abraded, so they'll come up even though the area has not burned. One of my more interesting herbarium finds were about 10 specimens that actually had seed packets on them. These seeds were 78 to 88 years old, so I wanted to do some germination tests on these to see how many of these would actually still germinate. Studies were done in the 1960s where seeds that were about 50 years old were looked at. That's the oldest ones that have been tested before this study, and they did have some germination in the oldest seeds. Here you can see a comparison of percent germination from the 78 to 88-year-old herbarium specimen seeds to more recent collections. Note that seeds from some specimens had 100% germination that were 88 years old. All test groups had at least some germination. Note the mean percent germination is much lower for the older specimens. Whether this is due to them just being older or being stored in an herbarium under unknown conditions is not known. Outside of burn areas, I have personally not seen any germination of Malacothamnus. However, sprouting by rhizomes is pretty common in certain taxa, particularly along coastal taxa. Sprouting by rhizomes may be an adaptation to help plants move into areas of less competition. One thing that's unknown in areas where plants are cloning by rhizomes is how many of those plants actually are clones versus ones that grew from seed. In many areas where plants grow from seed, you can have thousands of individuals. Do these individuals mostly die out and get replaced by clones? Unknown. This can be particularly problematic in assessing how many plants actually are in an area. So why study Malacothamnus? There are currently 3 conflicting treatments in use for the genus. The Kearney treatment on the left has the most taxa. This is the treatment mostly followed by MUNS and the California Native Plant Society Rare Plant Inventory. In the middle is the Bates treatment, which recognizes the least number of taxa. This is the treatment from the 1993 Jepson Manual and the Flora North America. On the right is the Slata treatment. This is the most recent treatment, which is somewhat intermediate between the other two. This is the treatment from the current Jepson Manual. Where treatment choice becomes important is in regard to which taxa are considered rare and protected by state and/or federal listings. Here I've highlighted in yellow those taxa that are listed as rare in California by CNPS. In gray are taxa that would not be considered rare in that particular treatment. Let's take a closer look at Malacothamnus fasciculatus as recognized by Bates. On the right, I've mapped Bates' concept of fasciculatus, which is one variable species with no subtaxa. On the left, you can see that Kearney recognized the same group as 9 different taxa. 6 of these are listed as rare plants by CNPS. One of these is listed as both state and federally endangered. Here, I've circled in red those populations that are currently protected by some form of listing. Under the Bates treatment, none of those populations will be protected. They are all considered the same. If Bates is correct, maybe this is okay. In this case, those taxa could be delisted, and time and money that is spent on them could go to other taxa that possibly need it more. If Bates is wrong, though, it needs to be made clear which populations are different, which need protecting, and how they can be distinguished from each other. The primary goal of my research is to answer those questions using the combined evidence of morphology, DNA, and reproductive barriers, such as flowering time and geographic isolation. I'm going to mainly focus on morphometric analyses here, as that is my current research priority. I've measured about 40 morphological characters on almost 800 specimens, representing the full morphological variation and geographic range of the genus. One of my primary tools is principal component analysis, or PCA. If you aren't familiar with this, it essentially reduces the dimensionality of the characters measured so that you can visualize them all in a single plot. This is used to explore data for patterns. On the left is an example of PCA with 3 distinct clusters. On the right is an example with no clustering based on the groups that have been assigned. The first group I want to show you is the Malacothamnus palmeri complex. These are the only 3 taxa in the genus to generally have capitate inflorescences. Bates considered these a single taxon, whereas Kearney considered them 3 varieties of the same species. The Malacothamnus palmeri complex occurs just in Monterey and San Luis Obispo counties. In the PCA, you can see that the varieties of Kearney form 3 distinct clusters. These 3 taxa are morphologically distinct with no intermediates. You can see on the map that they are geographically isolated as well. The lowest red circle on the map is a questionable location. DNA evidence also supports these 3 taxa. Based on the full evidence, I'm bringing these 3 taxa up to the species rank. This study will be in the next issue of Crossosoma, so watch for that. Taking a look at characters to differentiate these taxa, you can see on the right that variety Lucianus has much longer stem trichomes than the other 2. This character easily differentiates it from the others. This slide shows stipular bracts on the top and leaf pubescence on the bottom. Both are also quite distinct between taxa. A similar problematic group is the Malacothamnus jonesii complex, which is treated as a single species by Bates and 3 species by Kearney. These primarily occur in San Luis Obispo County. You can see based on the map that the ranges of these 3 taxa aren't quite as distinct as that of the palmeri complex, and that there's some of the plants appear to be both morphologically and geographically intermediate. 3 taxa do form 3 distinct clusters in the PCA, with some plotting intermediately. While these 3 taxa are mostly morphologically and geographically distinct, they can't be distinguished in the preliminary DNA analyses. This is perhaps a good example of taxa that should be recognized as varieties of a single species. They're clearly diverging, but maybe aren't quite diverged yet enough to be treated as distinct species. Further DNA analyses may help with this decision. You can see here that Malacothamnus niveus is easily distinguished from the others based on the much longer and denser calyx trichomes. Here you can see that Malacothamnus jonesii is easily distinguished from the others based on having a spike-like inflorescence. The others can occasionally have spike-like inflorescences, but in those cases, there should be plants with panicle-like inflorescences growing nearby. A major point of confusion in the genus is Malacothamnus orbiculatus. Bates considered this part of the Malacothamnus fremontii, but both Kearney and Slaughter treated Orbiculatus as a separate species, which is confirmed by DNA. While some plants of fremontii and Orbiculatus look almost identical, I found that they can be told apart fairly easily based on how the flowers dry, as pictured in the top right. The clade, including Malacothamnus fremontii, have flowers that dry at least somewhat open. Malacothamnus orbiculatus and most of the rest of the genus have flowers that dry closed. You can see on the map that they have distinct ranges that don't overlap. There is, however, possible hybrid zones shown in yellow. One intriguing question about the range of Malacothamnus orbiculatus are the disjunct populations in Arizona. These are fairly recent discoveries that have been mistakenly called Malacothamnus fasciculatus. Examination of specimens and photos show that they look much more like Malacothamnus orbiculatus than fasciculatus. Being so disjunct, these were clearly candidates for analyses. Are they potentially an undescribed taxon? The answer appears to be no. The morphological variation of the Arizona population fits completely within the variation of the California populations based on the characters I measured. Likewise, DNA places them in the same clade. The Arizona plants probably represent relictual populations, indicating that Orbiculatus possibly was more widespread in the past in what was currently the Mojave Desert. The question that started all this research revolved around Malacothamnus aboriginum, which is mostly from San Benito County, shown in purple. Bates believed there were also populations of this in San Diego County, shown in yellow. However, these plants wouldn't key out. Tom Chester and I decided to take on this question of what the San Diego plants actually were. Initial examination of herbarium specimens showed that San Diego aboriginum looked much more like its neighboring species, Malacothamnus fasciculatus and densiflorus, than it did the actual aboriginum. So we chose to focus measurements on these 4 groups. Here you can see that although some specimens plot somewhat intermediately, the 4 groups form 4 distinct clusters. Pairwise comparisons showed all 4 groups to be distinct from each other. Based on this evidence, Tom and I described what has been called aboriginum in San Diego County as the new species Malacothamnus enigmaticus, which was published in Madroño last year. Preliminary DNA evidence confirms that enigmaticus is much more closely related to the other San Diego County species than aboriginum. Note here how Malacothamnus aboriginum has much wider calyx bracts than the rest. Enigmaticus has calyx bracts similar to densiflorus and trichomes similar to fasciculatus. Here, each of the squares represents 1 square millimeter of a stem. You can see that the stem trichomes of aboriginum are huge compared to enigmaticus. One thing enigmaticus and aboriginum do have in common are relatively wide stipular bracts compared to the other 2 species. So they do at least have one thing in common. I don't have time to show you all the groups today, but just for fun, here is a PCA of everything I measured so far. There's too much on here for all the groups to sort out on the first 2 principal component axes, but a few clearly do. The 2 most distinct groups on this plot are Malacothamnus palmeri variety lucianus and Malacothamnus densiflorus variety visitus, both being distinguished by having much longer glandular trichomes than most of the rest of the genus. I still have a lot of work to do. The current goal is to complete the morphological analyses I'm doing to clarify what the morphological groups are and how to tell them apart. We'll be assessing each of these groups using DNA analyses, which will hopefully clarify the relationships between all the groups. Likewise, it may reveal cryptic taxa within morphological groups. Once all my analyses are complete, I plan to publish a treatment of the full genus with keys and illustrations. This will include at least a brief conservation assessment of each taxon. With that, I'd like to thank the huge number of people who have helped with this project so far, including the people and organizations that have helped fund this research. I'd especially like to thank people who have contributed observations. These have been critical to my research. You can see here that over 1,600 people have contributed over 4,500 observations of Malacothamnus to just iNaturalist. Many other observations have been contributed through CalFlora or via email. I'm still happy to accept any observations, especially from areas that are undermapped. I prefer observations posted to CalFlora or iNaturalist, ideally with good photos of various plant parts. I monitor these websites, so I should see them if you do post them. My email address is below if you want to contact me. My website is also below, which does have some Malacothamnus information. I already have some draft Malacothamnus keys posted there. I'm hoping to improve those keys over the winter so that people can test them over the next season. Please use these keys and give me feedback about what works and what doesn't. It would be great to fix any problems I can before the keys are published. Thank you very much. And now for our bonus feature. Abraham Lincoln Trichome Model. While measuring an excessive amount of trichomes on almost 800 herbarium specimens, I made the sudden realization that 0.5 millimeter was starting to look pretty big to me. I wanted to put this into perspective by comparing these trichomes to a common reference that at least most Americans are familiar with: the Lincoln penny. I had initially thought that I'd use Abe's head as that is such a familiar object, but alas, my trichomes were pretty darn small. While very tempting to illustrate these trichomes as Abe's nose and ear hairs, I decided little Abe on the back of the penny may be a much better trichome model. Little Abe is rather unadorned, so very suitable to play dress up with. First, let's look at the glandular trichomes on the calyx of Malacothamnus gracilis, shown on the right in red. These are approximately 0.3 millimeters and are on the smaller side of what I consider fairly long glandular trichomes in the genus Malacothamnus. These shall be Abe's talons. They're a bit large for fingers, and only 3 fit on each hand. Abe forgives me though. He's having fun, and is happy to step outside his usual box for educational purposes. Here I've shown some of the smaller glandular trichomes in Malacothamnus. On the lower right is a glandular trichome on the stem of Malacothamnus aboriginum. These are 0.03 millimeters. That's 1/10 of the size of the previous glandular trichomes shown. Zooming out quite a bit on the upper right, you can see these are dwarfed by stellate trichomes. The glandular trichomes here are the little nubs sticking up at the bottom of the photo. They're also Abe's toes. No foot talons for Abe. Looking at the stellate trichomes on the stem of the same species, you can see they are much larger. The rays of the trichomes, that is each part that sticks out, are about 0.5 millimeter long. This is fairly typical for Malacothamnus with longer stem hairs, though they can be much longer. Here Abe models his stellate trichome in defiance of his famous top hat look. Here we see the smaller side of stellate trichomes. The stellate trichomes of Malacothamnus fasciculatus variety Nutalii often have rays averaging around 0.1 millimeters. Here Abe models these stellate trichomes on a Hawaiian-style shirt. I didn't do any pollen measurements, but it seemed fitting for Abe to have some cool sunglasses to go with his shirt. The spiky ball on the right is pollen of Malacothamnus davidsonii, which is typical of the entire genus. This is 0.06 millimeters in diameter. On the larger side of glandular trichomes in the genus are the stem trichomes of Malacothamnus palmeri variety lucianus. These could be a whopping 1.4 millimeters in length. Abe holds one of these gigantic trichomes for your viewing pleasure. Now to put this all into perspective, let's zoom back out on the full penny. You can see on the right, Abe and his trichomes are barely visible. Finally, on the left, you can see the very long rays of the stellate trichomes on the stem of Malacothamnus palmeri variety lucianus. These are some of the longest trichomes in the genus, reaching as much as 4.8 millimeters in length. This is illustrated by the horizontal white line on the penny. Only a few species in the genus have trichomes this long. Thank you for joining us in our Malacothamnus trichome fashion show, and special thanks to Abraham Lincoln for being such a great trichome model.